Frederick. So
I mean, I learned a lot, but it was
the NIH library.
Yeah, just because
like it's like consistent.
Yeah.
Anyways,
Yeah, a lot of I agree. It's very
like my professor wanted to be huge because as possible. Talking wanted
last week we had
like the opposite of that. Is write everything out.
Oh, yeah?
I didn't say it ahead of time.
stuff like that. They'll organize a
Yeah.
Once goes like,
Frederick is where
you know, I was
the
just doing that.
It's what?
Frederick is, I think, where the mouse genome people were.
Right, we get it.
Nancy Jenkins and Neil Copeland were.
We should have this one.
Yeah.
The one thing that's mental is
You don't need to. You should.
Right. Is that the cue for me?
I did get it.
Oh, well,
Before you start, I'm going to say a couple
I figured,
words.
yeah, do this.
Sure. I just want to remind everyone this is the last of the semester.
this is the last.
Oh,
No more free pizza.
Don't
show up,
remind
don't show up
everyone
next
right
week
now.
expecting lunch. You'll be
Like
hosed.
a cake or something.
So you're allowed to take your stats class next semester, like the series.
So I don't have to take a series, I just have to take one class for the master's. And then like stats isn't even required at all for DCMB, which I think is kind of ridiculous, but
Why?
because I feel like everyone should take stats, like you're gonna do stats at some point.
What you said in the
Right.
context.
Like, I feel like the program has to require it.
Yeah,
You don't know the right
so
context, you
I wanted to take it
misinterpret
anyway.
the data.
And pass around the sign-in sheet. Yeah.
Context, are they?
Nope.
So yeah.
Sometimes humans are not even better.
Wow, that's especially down here. Sorry, you got me going. I know I can contact you, but this,
Yeah, I know.
you're the one I want to anyway. Yeah, you guys.
You want to go ahead and start?
Ready.
Nope, I'm okay.
So we'll go ahead and get started. I just want to remind everyone that this is the last seminar of the semester. We will start up again in the fall, and of course I'm looking for speakers, so if anyone is interested and willing to talk, please let me know. And I think I'll let Steve do the introduction.
Yeah, I don't think I need a microphone. I don't even know if this is on. Is it?
It's for the recording though.
Yeah, it's for the recording.
Oh, it's for the recording. So is it on?
It's on.
It is. Okay. Yeah. So it's a pleasure for me to introduce Ricardo Albanus. He is a third-year PhD student in the bioinformatics program here at the University of Michigan. Started off doing a rotation at the end of his first year and has been working in the lab ever since, and has made some nice progress on a project he's going to tell you about today, where the idea is trying to identify in high resolution how transcription factors interact with DNA, specifically using an assay called a TAC-seq. The projects he'll talk about are unpublished so far, although we're working on writing it up now, and I think he's even going to share you the GitHub URLs where this code is open and available for anyone to download and use, and I'd advocate for people to give it a try out. Especially, hopefully, you're interested after you see some of the performance metrics that Ricardo will show relative to other competing software programs. Finally, at the end, he'll introduce kind of a more provocative idea of measuring transcription factor residence time on DNA using accessibility data only. So I'd encourage you guys to speak up and ask questions, especially at this stage of the research, right before we're going to submit for publication. We would appreciate any kind of feedback you have. So thanks for coming in. All right, Ricardo.
All right. Thanks, Steve. And book something. No. All right. Can everyone see the screen from here? Okay, good. So, as Steve said, I'm going to talk today a little bit about chromatin accessibility and how we can use this kind of data to predict genome-wide transition factor binding and, in the end, residency time. So, to motivate this a little bit, we know that the majority of disease and trait-associated genome-wide association studies, the genetic variants are located in non-coding regions. So that means that rather than breaking, altering protein structure, they are affecting some regulatory measure in the cell. So
Oh, gee was, okay. So I think the statement is true for common diseases. I don't think it's true for mendelian diseases. the majority of Mendelian disease-associated variants are in the
Yes.
coding regions in the exome. So I think it's
Yes, for the
GWAS is common disease, right? So it's kind of like,
GWAS is the method. I mean, it's implicit in the
Yeah.
statement,
Yeah.
but GWAS is how you do common diseases.
yeah, you can't see rare stuff in GWAS.
okay, yeah. And for Mendelian, normally you have some sort of transcription factor being directly broken, and that's why the effects are so high for a single variant. So we can see that those the GWAS variants, they're enriched in regulatory elements such as open chromatin region or known transcription factor binding sites. So this shows their regulatory role, and also suggests that they may disrupt this regulatory architecture by disrupting transcription factor binding sites. So I'm showing here is the stretch of chromosome 8, where we have at the start here this RS508-419 SNP, which is linked to ANK1, this is a gene expression, and to type 2 diabetes. And you see that this region closely resembles this TR4 binding motif. So TR4 is a transcriptional repressor. And this SNP falls right in this position A, where you have, position 8, where TR4 is expecting to find an A in the binding site. And you can break so looking at what the non-risk and the risk alleles are, you can see that A, the non-risk, you leave the motif intact, but for G, the risk allele, G is not expected in this TR4 motif, so TR4 won't be able to recognize, and then you would disrupt the binding site here. And we have data showing that this is exactly the case. So ANK1 expression is higher in the risk allele, so the transitional repressor is not binding, and therefore is not repressing the gene. And this is probably the mechanism by how this SNP is linked to type 2 diabetes. So being able then to predict where those transcription factors bind in a given condition can be a powerful tool for uncovering the effects of these non-coding variants. So essentially we're trying to understand the underlying regulatory grammar of the cell regulatory architecture. And the go-to assay for this would be ChIP-seq. So you can assay a transcription factor, the transcription factor binding events using this technique. But it has some limitations, and the main one is that you can only assay one transcription factor at a time. So if you're thinking of all the possible 1,300 known transcription factors in the cell, you're going to do 1,300 sequencing assays, which is not practical. So an alternative approach is to infer transcription factor binding using chromatin accessibility data. So by chromatin accessibility data, we'll be talking mostly about ATAC-seq, so this is the assay for transposase accessible chromatin, which infer accessible regions using transposition. So it's very similar to DNase 1, but instead of having DNase 6, but instead of using the DNase 1, you're using a transposase that's already loaded with the Illumina adapters. So you can use less input material. So instead of millions of cells, that's what you need for DNase-seq, you can do it with thousands of cells, and the readout is essentially the same. Okay, so, okay. All right, so the question we're trying to answer here is, can we accurately predict bound motif occurrences using this chromatin accessibility data? So I'm showing here this IRF8 locus, where we have different tracks here. The track number 1 in orange is the GM12878 ATAC-seq data, so the chromatin accessibility. You can see that it varies across this locus. And then on 2 here, we have the ATAC-seq peaks for the peak calls for this region. You can see those regions with high chromatin accessibility got called as peaks. And then we have this SP1 motif. So these are regions where the sequence match the recognition site for SP1. And we also have, for this region, the ChIP-seq data for SP1. So we know where SP1 is binding. And you can see that there are four or five motifs here that are bound by SP1. So the question is there any feature in this orange track that we can use to predict which of all these SP1 motifs are going to be bound, and not only for SP1, but for all the other transcription factors. There are a couple methods to do that. The most widely used ones they are the footprinting-based methods. So they are searching for regions of relative protection on open chromatin. So what does that mean? So in panel A here, I'm showing this MTPN locus, and I have the ChIP-seq data for NIF1. So this is another transcription factor. And right below, I have the DNase data, so the chromatin accessibility for the same region. And you can see that there's an overlap between those two peaks. So this is the smooth signal. So we actually zoom in the cut, the cleavage pattern on this region. So that's a white box here. You're going to see that we mostly have a high density of cuts, but right at this locus here, we see this depletion. So this is what the footprinting methods are looking. So the assumption is that NRF1 is sitting at this region and is protecting the DNA from cleavage, right? So those footprinting methods are looking for open chromatin, a dip in the signal, and then open chromatin again. Then there's also this raw signal approach, which is the one we're going to discuss more in depth. So it has been known that if you just sort your motifs by the density of cleavage or integration events in the vicinity of a motif, that's actually a good predictor of whether that motif is bound in the ChIP-seq data. So I'm showing here the CTCF motif. On the x-axis, you have the relative position for the motif, plus minus 50 base pairs, and the colors indicate low to high DNase cleavage, right? And if we sort by the number of cuts in this on the y-axis, you're going to see that there's a good correspondence. So the most occupied motifs by ChIP-seq will also be the ones with highest cleavage. Okay. Okay. So there are already a couple of methods that do that. I'm showing here data from this Guzmão et al. Paper. Is this method called HINT. This is one of the most recent methods that we have. It uses this hidden Markov model for looking for those dips in the signal. And this is a figure from the paper where they compared their method to a bunch of other available methods. And on the x-axis here, we have different performance metrics and the ranks for those methods. And their method they found was the best performing method compared to all the others. There are a couple of caveats if you're thinking of this footprinting approach. So first of all, these footprint-based methods, they only look at regions within chromatin accessibility peaks, right? So is this an important limitation? Do all factors, the bound events, are in open chromatin? Second, more important, do all factors leave footprints? Are you going to see that depletion in signal for every single factor? And then if yes for both of those, if no for both of those, is there a better, more general approach? Okay, this is not working. Okay, so answering the first question, do all factors bind in open chromatin? This is going to depend on the factor. So I'm showing here this bar plot on the x-axis I have different factors, and on the y-axis I have the fraction of bound motifs, so ChIP-seq bound motifs, relative to ATAC-seq peaks. So this is looking at the Buen-Russell GEM 12878 data. And I'm showing in blue the fraction of bound motifs that are within ATAC-seq peaks, and in red the fraction that's outside ATAC-seq peaks. And you can see that for some factors like BRCA1, 100% of the bound events are within open chromatin regions. But other factors such as EBF1, SRF, almost half of them are outside. So if you're only looking within peaks, you're essentially missing out all those red parts of the graph. And to my knowledge, unless you do this experiment for every single factor, there's no way to predict whether your factor is going to be binding outside peaks or within. Right? So this could potentially be a limitation.
That strictly bind proteins, so they're part of a complex and actually don't bind the DNA at all?
Yes. So
You kind of miss those entirely,
yeah, but this is a
or maybe by proxy through something else.
yes. Yeah, this is a limitation, but I think it's for every single one of them. Unless, yeah, then you would do a chip seek for those guys, but footprinting you won't be able to do unless you know what is the motif of the cofactor in question. But yeah, this is
One more question to this one. You show the dependency on the factor. What about tissues?
sorry.
Dependency on tissues? I don't think it's the same for every tissue for every factor.
Yes, you're going to have different factors active at different tissues
So
too.
that's a tissue-specific profile.
Yes. So we have data for another GM20878 attack-seq sample, and we pretty much see this pattern, but I didn't run that to another tissue. Yeah, have to look at that. That's a good point. Tissue. Okay. So next, do all factors leave footprint? And apparently this is not the case, or at least it changes between factors. So this is from a Gordon Hager paper from Gordon Hager's group, where they're showing what is the amount of protection for different factors. So for these three factors, CTCF, AP1, and glucocorticoid receptor, they are showing on this, there's essentially the plots here are showing the motif at the center, right? A 30 base pair region on each side of the motif. And then they have the cut count, so how much cleavage events do you have for those motifs, right? And they're showing what's the difference. So the first horizontal dash line here is the average background on that motif, and what is the protection at the motif, right? And so the further away those lines are from each other, that means the higher the protection is by that factor. So more of a signal depletion it's causing. And they found this, it seems that's correlated with residency time. So factors that bind longer to the DNA, they live deeper, they protect the DNA better than factors. So glucocorticoid receptor is known to bind for a second, 10 seconds, sometimes maybe less than a second, and then you cannot even see a footprint very well, whereas CTCF is known to bind for minutes at a time, and you see this really nice protection. And so this is a limitation, not all factors leave footprints.
So how do people know how long does it bind?
So I'm going to talk about that, but you can only do that through experimental assays. So you can do that through single molecule tracking. So you design a GFP tagged version of your transcription factor, and you look at the microscope and see how long does it bind the DNA. And there's this photobleaching assay, which I'm going to describe later on, that gives this information. But still, it's experimentally complex because you have to design a GFP tagged version of your factor, express that into a cell line. So it's not, you cannot do that for every factor.
So why the baseline are different? I mean, the baseline are different. Why that?
Oh, you mean, yeah, here? So that's a good question. But it depends on where those factors are binding. So AP1, for instance, is binding at promoters, right? So you'd expect to see a higher baseline just because promoters are more open chromatin than, for instance, the glucocorticoid receptor. Those are known to be pioneer factors, so you should be binding at regions that were previously closed, so the baseline would be lower. And CTCF, it's its own environment that's also very open chromatin.
So just to add to that, you're absolutely right, but also in this plot, they're looking in pretty close proximity to the motif, right? Plus or minus tens of bases. If you were to zoom out and look hundreds or kbs away, then they would all come down to exactly the same baselines.
Does that mean it's because when you are mapping, you might have some ambiguity and make the baseline of that smooth?
So mapping bias?
Yeah.
Yeah, so, well, if you answer that.
So I didn't understand the question, sorry, can you repeat?
So according to Steve's explanation is because we are just looking at a very small region.
Mm-hmm.
So when you see a lot of fluctuation in a small region, is that because you have some bias in your mapping?
Yeah, so they account, that's actually one of the things that those methods try to account. So if you look so this is an aggregate plot. So you're looking at here 40,000 motifs, 12,000 motifs, 6,000 motifs, right? So with all of them, this region at 0 is essentially the same, right? Because it's the same motif, but the region around it's going to be slightly different. And you see, so you see there's like peaks here for glucocorticoid receptor. This is the integration bias of the technique. So sorry, the cut bias for DNase, right? So these models actually try to see what are the preference for DNase binding. So they do like DNase on a naked cell, so a cell without a deproteinized cell, and try to model what are the preference and take that into account for the model. But still, you cannot open too much the region that you're looking at on either side, because you can also have other motifs close. So if you look at promoter, there are several motifs co-occurring, right? So if you start opening too much, then you're already looking at the motif next door. So it has to be close by. Yeah. But those methods, they try to take into account this DNase bias, the mapping bias. Not mapping, sorry, the cleavage bias. Cool. All right, so we had some issues with the way they compare the data in that hint paper, and we decided to look at ourselves. So we compared 5 different methods on ATAC-seq data. That's an important point. That method was compared in DNase data. We're looking at ATAC, which hasn't been done before. So GM12878, and we're also including human pancreatic islets, so looking at clinical samples. So we're analyzing that hint paper, so the 2016 one that I mentioned earlier. DNase to TF, that's the paper from the previous plot. Centipede from Roger Pichireggi, who's currently at Wayne State next door here. So Centipede is not a footprinting method. It's actually an unsupervised clustering method. So it looks at the pattern, the cleavage pattern around each motif, and try to separate between bound and unbound motifs using this multinomial model. So it's not dependent on looking at within peaks. It's a motif-based approach. Centipede partition, which is a different implementation that we use for the Nature Communication paper, that takes, besides taking the cleavage pattern around the motifs, also take the fragment sizes. So if you have pair-end ATAC-seq data, you can also include the fragment size into this. And finally, this new signal-based approach that I'm going to explain.
Question. So, how is it conceptually different that these methods that you list in your method from other methods that predict transcription factor binding, like DeepC or ThanQ, that are, deep bind, all of those things that recently came out in literature, and there's visiting speaker, a couple of last weeks that gave a talk about this. So like all the methods that they use, DNA sequence, some other maybe information to predict if it binds or not, and they use ChIP-seq for validation. So how footprinting and then what you do, is there a conceptual difference in the task you're performing versus what they do, or is it exactly the same?
No, it's not the same because, at least in my understanding, those methods are supervised. So you need to train them, right? And we're not sure exactly. So if you train on CTCF data that you have plenty of CTCF data sets around, maybe you'll be very good at predicting CTCF. But this is not generalized. You don't know how that's going to work. Say, for instance, if you want to use a CTCF model on a FOXA2, right? So all these methods, they don't need any training. So you can just take your sample and straight up give to them. You don't need additional data. So if it's a cell type that you don't have other ChIP-seq data, anything, you can straight up do those techniques, and we're going to see how well they perform. That answers?
Yeah, so the interesting part that we're actually writing the review paper right now, and the interesting part is, for example, DeepBind, they show that the method can generalize between different transcription factors and between different cell lines. So you can do like a transfer learning kind of part. So if you train on a number of transcription factors on a number of cell lines, you and use the same model to improve your predictions in another transcription factor in another cell line. So that's interesting, sort of, there is something common between those different transcription factors and different cell lines. There is some common information that can be reused again. But of course, if you can achieve that with unsupervised approach, that's much better.
So my guess is that those methods are learning the raw signal, and that's what we kind of see with Centipede. It wasn't actually learning a lot of patterns. It seemed that it's just learning the amount of signal on those motifs. So that's definitely something that we can test in the future. But right now, I'm just going to focus.
What's the actual input data when you're using that? The sequences. So you don't know if the transcription factor is present in a particular cell type necessarily. This is independent of that. It's saying things were bound because they're interfering with the integration of the transposon. So you actually have data that's looking at the state in whatever cell type you're looking at. And it's not for specific transcription factors. It says, is a protein bound? It is conceptually different.
Yeah, and I guess I would add to that, this transfer learning, I think will work reasonably in a few instances, but I don't think it's going to be broadly applicable to lots of different TFs. For example, Terry Fury's group just published a paper in NAR about their method called DEFCON or something. It's kind of a fun little name. Where they do transfer learning of TF in one cell type, and they perform very well in the same TF across another cell type, right? So you could imagine TFs generally behave the same across generally, right? Not very specific instances. But all these 1,700 TFs in the human genome, some have very similar protein domains, others have very different protein domains. So I don't think transfer learning from one TF is going to be applicable to another TF unless they're very similar, like paralogs or something. Right? So I'm skeptical of that kind of.
DEFCON 1.
Just DEFCON.
You can choose the number. All right. Okay, so let's see. Those are fancy methods. Can we come up with something simpler that you don't need a PhD in machine learning to run, and even if you say that they are user-friendly, they're not, right? So at least anything that they say it's easy to use, to my experience, is never easy to use.
You should drop your guard.
Yeah, exactly. Oh yeah, this is going to be easy. But okay. So how is this raw signal approach that we're talking? Essentially, we just count the number of fragments that are occurring in the vicinity of the motif. With the fancy part of the model is that we just discard the fragments that integrate directly on top of the motif. So because there's sequence bias here, that's the only part that's common between all the motifs, we are discarding those. So I'm showing here two CTCF motifs, right? And then we have this extended region plus minus 100 base pairs. So the raw signal score for this guy here would be 16. So there are 16 tags occurring in the vicinity. You're discarding these two. And then for this other, the raw signal score would be 6. And then we just rank them by that number and see how they perform. All right. So how do we evaluate those predictions for all those methods? We have, so this is GEM 12878. There's this richness of encode data. We have chip-seq data for several factors. So we intersect the motifs with the chip-seq peaks, and we classify those motifs as true positives or false positives, right? So if they're bound or unbound. And then we look at the method prediction, so saying, is this motif bound? Yes, no, no, yes. And we label them as true positives, true negatives, false negatives, and false positives. So essentially the data that we have, for each method we have this table where we have the motif prediction score rank, so the p-value or whatever score the method outputs, and whether that motif is a true positive or not. So a good method would have a lot of true positives early in the list, and a not so good method wouldn't be able to discriminate between the two positives, so they would be evenly distributed across this list. And then we can evaluate this list, using either raw curves or precision recall curves. So raw curves are plotting the true positive rate versus the false positive rate of your data set. And then I'm showing here for each of those methods. So this is ELF1 on when Russell Jam 12878 on all regions, so not only within peaks, we're doing genome-wide. And you can see that we can take the area under the curve of these raw curves, and this is going to be a metric of which method is better than the others. Then we have ranked here raw signal, 0.97, centipede non-partition, 0.96, and then last hint and DNase to TF. But since those are unbalanced data sets, which means you have a lot more true negatives than true positives, we're going to focus instead on this precision recall curves. So they're giving at any particular recall. So if we look, once we get 20% of all the possible true positives in the data set, what is the precision that we're calling them? So let's say for DNase to TF, when we find 20%, so as we go down that list and we get 20% of the true positives, what is the precision? So that's around 0.7. So that means that 7 out of 10 of everything we're calling is true. So the precision. But for raw signal, let's say, at 20%, that precision is 90%. Is 90, right? So 9 out of 10 are real. And then you can imagine a perfect method would do 100% of the recall with 100% precision. So it'd be shifted to the upper right here. And again, we can take the area under the curve and directly compare those methods. So I have here, based on the precision recall you see, raw signal is the first, followed by centipede, hint, centipede partition, and DNase to TF.
With these calculations for precision and everything else, is it true that you use the ChIP-seq data as the gold standard?
Yes.
Only?
Yes.
Which means you optimize to the systematic errors of ChIP-seq, not to the truth.
Yes. So.
And ChIP-seq has a huge error rate.
That's true.
Wouldn't it be better to have something more reliable than ChIP-seq as a gold standard and do the same thing?
I'm open to suggestions on that. So what could be an alternative?
Well, a combination of ChIP-seq with some of the other methods that you showed. And if you look at that, you would find a few more true positives that are missed in ChIP-seq simply because of the cutoff.
Mm-hmm.
And you would get rid of some false positives that are pile-ups of for whatever reason. And well, you've done this basically. But it's really, you're optimizing something to the error rate of ChIP-seq here.
Yes.
And that worries me a little bit because that's such an elegant approach. It shouldn't be based on a faulty method. And ChIP-seq is basically a faulty method. We just don't have much better. That's the only point.
I think this is a great point and something that I discussed extensively with Roger Picchi-Reggi and other people. And this is a problem of the field as a whole. I don't think, I haven't seen any discussion on what could be. I see a lot of people complaining exactly about.
I would not replace one faulty method by another.
Mm-hmm.
Because then you're just optimizing to a different error. I would try to compensate the error. It's a very simple thing. If you have an error rate of 10% and you use one method, you have an error rate of 10% to which you're optimizing. Mm-hmm. If you have 10 methods with an error rate of 10%, in the end you have 1% error that's really residing. That's the idea behind it. So a combinatorial approach for the gold standard. And it might be worthwhile investing a little bit into getting a better gold standard here, because everything that follows is closer to the truth.
Mm-hmm.
Just my 2 cents.
Yeah,
so my issue was sort of along the same lines. Are these, so the transcription factors are all assumed to bind by themselves in that, right? Because what I'm worried about, and if you go back one slide for this false positive, false negative, it is conceivable that there is a binding site for a transcription factor, and it's not a false negative prediction, false positive prediction, but that the transcription factor in certain circumstances, you know, there's a repressor that's preventing it from binding, or that they have to somehow interact. And there's no consideration of protein interaction at transcription factor binding sites right now.
No.
And we know that there are many transcription factor binding sites that are like 2 or 3 and acting cooperatively, or a repressor inhibits, and all of that is not in there, right? It's just assuming. So my guess is sort of modifying what you're doing is for certain transcription factors like CTCF maybe, that are very common and that bind just by themselves, this will be good. But for any complex factors, you may get too many false negatives.
Yeah, so these are all valid concerns. And I think of this method not as giving you a definite answer. This is, you get your sample and you just want to survey what's the binding landscape there. You don't, if you really want details on a particular factor, then you're going to go and do ChIP-seq or
create the problem. If you do your method and you say this is my prediction, this is based on ChIP-seq, fine. But if you start comparing to other methods that might not make the same mistakes, and then they are less good because they don't make the same mistakes, that's where the issue starts. And what Mark said is just part of that. And there's much more to that, which is subsumized in the ChIP-seq error rate. And that's all fine for a single method. But comparing methods on a standard that is off the truth is unfair. Because if another method is closer to the truth, in your comparison it would fail. So it would be better in reality. And that's whenever you do a comparison, you have to be extremely careful about your standard.
So I think all this is great feedback, but can you forward to the next slide?
Okay.
You know, the current state of the field is ChIP-seq is about as good as you're going to do unless you go to ChIP-XO or something a little bit more high resolution, right? And I agree, ChIP-seq is not a great assay, but it's kind of the workhorse that we have today. And all of these methods that he's comparing against are using the same input data. It's all motif scans and all chromatin accessibility, right? It's just different ways of analyzing motifs and chromatin accessibility. So given the imperfections of ChIP-seq and that all of these methods are using the same input data, if those curves are shifted in one way or another, I would think that that is meaningful in terms of having better performance. So some of these things we can actually go back into the lab and validate, and we've dug deeper into the ChIP-seq data and found, yeah, this is an example. The ChIP-seq data just looks really, really crappy, right? Or this is a false positive unnecessarily because we actually see signal in the ChIP-seq there, just didn't meet their threshold for calling something, right? So there's all these caveats that we've seen and observed. But I want to, there's like, you're getting near the end and it's not even halfway through yet. So let's just finish up this discussion. No, yeah, it's just a great discussion, but we want feedback everywhere.
Finish up. So I think that that's a great point brought up by Stephen. The same refers back to what I was saying about the transfer learning by pushing these curves up and then matching your whatever ChIP-seq is, even if it's not right, you're still picking up something that generalizes well. That means you catching an important concept that's below this biological processes, which is a good thing to do. So not having the gold standard is one thing, but then you still want to do better. And another thing, some papers also do, like this is directly to this slide, some papers also do comparison of methods, recall at a fixed FDR. So you fix your FDR at some particular level depending on
10%, yeah.
Because I think that metric is important for what you want to do after you do your assay. So what you want to do is like for downstream analysis, you want to have some kind of like confidence what's your recall of the fixed FDR. So in your experience, do you think that the area under precision recall curve is like the metric that everyone should be using to compare those, or you also report?
That's a good starting point, but later we're going to discuss the F1 score, and I think that's the metric you should be using, because then you're taking your cutoff into account and you're comparing directly what is called as true positive by one method versus what's called as true positive by the other, even though you might draw a line here for a signal and maybe here for hint. So that's what you want to compare, because those methods, they have defaults for where do you draw the line. So that should be what you're comparing. I'm going to show this data later. But first, so I get the precision recall you see, not for F1, but for all the factors. I'm going to go kind of quickly here. So first of all, let's look at when roster data within peaks, which is where all those methods are operating, except for Centipede. And I'm showing each point here So on the x-axis, I have the precision recall you see for those different methods, centipede partition, DNase, TF, and hint. And on the y-axis, we have the precision recall for raw signal. And points that are above the line means that the precision recall is higher for our method. And you can see that pretty much across the board, all those points are above the line, with the exception of those green square, which correspond to CTCF data sets. So CTCF is the only method that's predicted better by this other, sorry, the only factor that's predicted better by these factors, this method, sorry. Then if you look at all regions, you basically see the same regions. The distance is higher for hint now because it's missing all those binding events that are occurring outside peaks. Then we did the same thing, the same analysis using our own GEM-278 data, so comparing the same biological, cell line but a different biological sample. And then we see pretty much the same results. CTCF is the only one that's below the line, right?
Any idea why that is?
We're gonna, if we have time, we're gonna discuss, because I have to run to this. Then we also did that on human tissue samples, which is where we actually want to do. And then the distance is actually greater than what we saw. So we have two different islet cadaveric donor samples here on the road, and the different methods on the y-axis. This is ChIP-seq data from this Nature paper and Pasquale paper that came out a couple years ago. And unfortunately, it's not matched to our sample, but we pretty see all those points above the line, right? So, answering Alex's question, how do we establish the threshold for footprinting? We see that just by taking the score vector, it works. But how do we draw the line here? So, for each motif on this measure, this raw signal approach, we get all the raw signal scores. Then we randomly sample motifs outside peaks. So those are most likely to be unbound. So this is our null set. And on this null set, we fit the negative binomial distribution on them. And then with this negative binomial distribution, we can calculate the p-values for all the observed values in the data. And the bound motifs will be the ones that the adjusted p-value by Bonferroni correction is less than 0.05. Huh. All right, that's why I like using my own computer. So there should be a distribution here. I don't know why it's not there.
Maybe it comes.
Okay. Anyway, I was just going to show the sample data and the negative binomial, and you see that they work. Yeah, this is annoying. It should be there. But let's. Oh, I did. No. Yeah. Anyway, so we can. So the negative binomial is composed of two parameters: the mean and dispersion, right? And so this is the mean and dispersion. So I'm doing a scatter plot of the distribution that we inferred for CTCF, and I want to look at how is this distribution similar to other factors, right? So there's this whiskers here saying we did this randomly sample. We fitted this distribution 150 times, so we have an idea of how those values behave. So let's see how it works for the other factors. And you can see that there's not much overlap between the negative binomial distribution. So this suggests we should be using a per motif negative binomial instead of a general negative binomial. And now we're going to compare that Bonferroni cutoff to the other methods. So I'm using this F1 score, which is a function of the precision and recall at that specific cutoff, comparing to the cutoffs suggested, so for centipede, DNase, TF hint, and also this within peaks. So just saying if a motif is within a peak, is bound, how does that perform? And you can see that the per motif negative binomial F1 score is higher across the board for all of them. And we can show the proportions here. So the percentage of time that the raw signal F1 is higher than the other methods, close to 90% on every method. So this cutoff is better than the others. So next, subsampling comparison. So all these data sets that we're looking are sequenced very deeply. So let's look at real data where we're not sequencing to 300 million reads. How much sequencing is necessary for accurate transcription factor prediction? Showing data here for ELF1 on the Brand-Warsaw data. this is just one of the samples instead of the pool data, and different sequencing depths here. And you can see that around 30 million, 20 million reads, it's already close to them. So I'm showing the precision recall AUC on the y-axis, and then as we increase the sequencing depth, we start making better predictions, right? So at 30 million seems to be already closing to the max. And we can look not only raw signal, but on the other methods. Centipede is kind of similar, but it's stabilized early on. And hint and DNase CTF, they're very bad at low coverage. So you cannot call peaks with low coverage, and they cannot look at the data, right? Because they're blind here. And then what we saw at 30 million reads is a good number for us. For them, it's almost 60, 50 million.
Does this vary by factor?
Does
this vary by transcription factor?
This is just for ALF1 here. And I'm going to show now for all the others. So we can rank those methods at different depths, and I'm going to show those rank plots. So at 60 million reads, which is close to the maximum of this data set, I'm showing the precision recall AUC rank. So 1 is the best, 4th is the worst for all the different factors. Raw signal in red, centipede in blue, and you can see that they are basically, raw signal is dominating the first place here, followed by centipede. And then hint and DNase CTF are basically competing for the 3rd and 4th place. If we look at 30 million reads, which is more like an actual sample that you would do, the competition is just centipede and raw signal, right? Just on the precision recall. And hint and DNase CTF, they don't even try to get to the 2nd place. So we should be using these approaches. And then if you look at the F1 scores, unfortunately I didn't have time to run this data for today, so it should be probably what we saw with the full data set with raw signal dominating the first place. Okay, finally, very quickly. So chromatin architecture information content. So can we predict residency time from ATAC-seq data? So Gordon Hager already gave some hints that those factors with high residency time leave these deep footprints. But how do we get this data in the first time? So answering your question, the most common method to get this is this fluorescence recovery after photobleaching. So the idea, so you generate this data set, this transcription factor with a GFP tag. And GFP, if you shine a laser on it, it's going to burn out, right? And so basically what you can do, so you can take your nucleus at the steady state level, and you burn out half of the nucleus, and then you count the time that it takes for the other molecules to go to the other side of the nucleus. And this is directly this is going to be directly a function of how long the factor interacts with DNA. So here I'm showing this time course from this Fair et al. Paper, where they have this HMGN1 wild type protein that binds to DNA. And you can see after bleach, it takes more than 20 seconds for it to go to the other side of the nucleus. But if you ablate the DNA binding domain of this protein, it takes less than 5 seconds for it. So it's just the diffusion speed of this protein, right? So this showing that it's directly dependent on the DNA interaction. And there's data for a handful of factors for this assay. So imagining a factor that binds for DNA for a long time, what would you expect to see in terms of chromatin architecture? So your factor would sit at the DNA. It's all wrapped by nucleosomes. So once it's sitting there, it's going to start shuffling nucleosomes around, because sitting there for a long time is going to give time for it to accommodate. So if you think of the accessibility landscape at this particular locus, the regions protected by the proteins won't be accessible to transposition, right? So let's put a y-axis here, that's fragment size, and let's plot how we would imagine seeing the fragments around in this locus. So we would see small fragments on both sides of the transcription factor before the adjacent nucleosomes, then slightly larger fragments on overlapping the transcription factor. Item color coding: blue is to the left of the transcription factor, black to the right, and red are the ones that are overlapping. And then as we go higher in size, we're going to see these mononucleosome fragments and even larger dinucleosome fragments, and so it goes. But we can represent this data just showing the fragment midpoints instead of the fragments. and we would see this sort of V pattern, right? So this is a high residency time factor that rearranges chromatin architecture. Do we see that for every factor? And the answer is no. Chromatin architecture apparently varies between transcription factor. So this is the same idea of that plot I showed before. It's centered at 0 at the motif center. On the y-axis, the fragment size, and each dot here, although you can see, it's a fragment midpoint. Right? And you can see for P53, there's no discernible pattern here, and we can actually measure the information content of this V-plot, and maybe there's a little bit of enrichment right at the middle here. But if you look at CTCF, there's a very striking pattern here. So the chromatin is rearranged around CTCF, and this matches that idea, those experiments that show that CTCF binds for a very long time, and P53 doesn't. So can we do that for all factors? And this is what I'm showing. So A here is just a recap of that schematic. And I'm showing here not only P53 CTCF, but CRAB3, which is an intermediary factor. And if we just sum up the information content around 100 base pairs, and do that for the factors we have, there's actually a very good correlation with the FRAP residency times. So factor CTCF is the highest FRAP recovery time, is also the highest chromatin architecture information content, and F kappa B is a low one, P53 is a low one, and you can see this. So now we have this metric that's a proxy of the residency time, and then we can start looking at other factors which you don't have these experiments.
By residency time, do you mean the percent of time it's occupied? Or are you talking about how long a molecule sits there?
Yeah, so that's a good question.
Because one is influenced by the concentration of the factor.
Mm-hmm. So there's a lot of mathematical modeling going on those FRAP assays. And the data is that the model we have, this is actual residency time. So there's single molecule tracking experiments showing that the factors with high FRAP residency time are also the ones that take longer. Like in a single molecule tracking, you see them sitting for a longer time. So it's not a measure, not a function of concentration, but the actual interaction time with the factor, with the DNA. Oh. All right. So we have this index, the chromatin architecture residency time score. So chart score is the sum of this information content around the motif. And if we plot the predicted residency time versus the precision recall AUC, we see that there's a positive correlation for all the methods. So this is saying all those methods, they are sensitive to factors with high residency time. Right? So this would make sense for the footprinting, because the footprint, they leave deeper footprints, so they are easier to predict. And you ask why CTCF was different. CTCF is the one with the highest information content that we have here. So it is bound for longer, and therefore the other methods can predict them. So if you just put all those lines together, you can see that the correlation is positive for everyone, but the intercept of this line is actually higher for raw signal. So even for factors with low residency time, we do a better job of predicting them than the other methods. And answering the question, is CTCF the rule or the exception in the transcription factor world? Is the exception. This is the highest information content. So all the other factors have very lower predicted residency time, so we shouldn't be using those footprinting-based methods for them, something else. Oh. In summary, even within peaks, raw signal outperforms or does as well as the other methods. The only exception is CTCF because of the high residency time. This is significantly simpler to implement compared to all the tested methods. I'm not going to go into details, but I mentioned already that they are not straightforward. It's just not a package that you download, input your data, and it just works out of the box. So the way we're deploying this raw signal approach is just a command line that takes a BAM file, a PIC file, a motif file, and gives the input the number of reads and the p-value of the adjusted p-value, and you can just straight use that. So no problem. Easier to run and troubleshoot. We have another formal benchmarks, but it's also faster and parallelizable compared to the other methods. relatively robust to samples with low sequencing depth, which is a problem for those footprinting-based methods because you need to call peaks on the sample. Still sensitive to residence time, although not as much as the other methods. And then future directions, we are still working on understanding which regions of the V-plots, or the blue, red, black plots, are predictive on the underlying chromatin architecture. Fortunately, I'm out of time already, but that score doesn't generalize very well across samples, so we're making it more stable so that we get the same number across different replicates, different biological samples. Also some metrics, some footprint validation quality controls using allelic imbalance at high information content positions. Also taking into account integration events at the motifs. Right now we're just discarding them, but we can actually use this metric as a quality control, and also using non-expressed transcription factors with non-motif as negative controls. And then finally start correlating structural domains with residency time. So now take that histogram and see what makes a factor have higher residency time than predicted residency time than the others. And then can't have tools and tags without tools. So won't have time to go that, but this is our GitHub, so Parker Lab, capital P, capital L. There's the AttackDK, which is a toolkit for working with attack-seq data. So right now the raw signal method is unpublished, but we have some tools that are already available. So the trim adapter is used for removing the Illumina adapters from the attack-seq data if you're doing that. Make-cut matrix is making the input data for Centipede, which is not straightforward to do. So we have a tool that does that for you. And then some quality control plot that you can analyze your data. And also the AttackUV, which is our quality control and visualization tool, which I hope it's going to work. Yes. Care about security. Yes. All right. So with this, you can look at your libraries. Where's the results zone? Come on. I'm trying to. I can't. All right. So refresh this. And you can see the fragment distribution of your samples. Everything is interactive, so you can like put your mouse over and see which sample we're talking about. What's the fragment length, the fraction of all the reads. You can see some nice metrics of distance from a reference distribution. So if is your sample over transposed or under transposed. Everything has its own help here. You can see the fraction of fragments at transcription start site. So this is a good metric of whether your sample is high quality or not. You can also get all the reads in peaks, peak territory, mapping quality distribution, all this stuff. Nice tables with the percentage of high quality autosomal alignments, everything that you might want to look whenever you get the data set for the first time. So everything is run locally. You don't have to send the data to our server, so you don't have to worry about us stealing your data or whatever. Anyway, and you can also send to collaborators so they can directly download the metrics or the experiments. So this is a companion to any attack-seq data. And with that, thank everyone. You guys for being here, and acknowledgements to the Parker Lab. Steve's great mentor, nice to work with. The lab's composed of John Hensley, our staff scientist, who makes everything works like clockwork. Yoshi Kiono, the postdoc, together with Jacob Kitson, that generated some of the attack-seq libraries. Arushi and Peter are PhD students together. Nandini Manikan, our lab tech, and also Laura Scott and Jacob Kitson for experimental and analysis support. So, open to questions, 5 minutes.
show.
Just a little comment on the last point. Your controls were non-expressed transcription factors and their binding sites.
Mm-hmm.
A great idea if you take into account that transcription factors are a little bit promiscuous. There are many transcription factors that can bind to the binding sites of other transcription factors. So you have to make sure that none that is capable of binding is expressed, not only one. Other than that, it's a great idea.
Yes, yeah. So we can derive some of this information from the position weight matrix from the ChIP-seq data. So we haven't started looking into that, but definitely you see some factors that the PWM is nothing. So it's basically binding anywhere, but others are have very strict binding requirements. So this is definitely going to be, we have to know these motifs beforehand.
Any last questions?
Nice closure of this
Yeah.
term.
Agreed.
Happy spring, summer term. Not that we all take off. We all are working spring, summer too, but
Yes.
I think today is the last day of classes, right?
Huntsman is Saturday.
Yeah.
That's great.
So I don't see you, Daniel.
No, I wasn't trying.